Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this section and following the biology syllabus, you should be able to understand and describe the principles of experimentation. What does this actually mean? What are we trying to understand? Well, it's straightforward enough. You have to be able to understand the characteristics of good experiments in science. Let's start by looking at experimentation or science experiments. The principles or guidelines that must be considered when carrying out scientific experiments involve having a fair experiment, a safe experiment, the experiment must be carefully planned, and the experiment must involve a control. We're now going to look at each of these principles and discuss them in more detail. Experiments must be fair. Usually this means having random selection, that the experiments can be replicated, it can quite often involve double-blind testing, and large sample sizes must be involved. What is random selection? Well, when choosing a sample to be tested, the selection should be random. So let's suppose I want to investigate the average height of a student in my school. Then this would involve choosing students in no particular order or pattern, that is, in a completely random manner. This is in order to avoid bias. Once this is done, the students I select should be representative, not only of a wide range of heights, but also a wide range of ages, body sizes, and fitness levels. In this way, the results, the average height of a student in my school, will be more accurate. If, for example, a certain type of sample is selected, perhaps I am biased towards the first years, then the results will be affected by that bias, and the average height of a student in my school will obviously be less than it should be. Another part of a fair test is the way that the experiment should be able to be replicated. A replicate is a repeat of an experiment. Don't forget, results are published in journals, magazines, on TV, newspapers, and on the internet, etc. So, others can repeat or reproduce the experiment to validate it. For those of you able for a bit more, when experiments are repeated, that generally refers to the fact that if I repeat the experiment myself, I keep getting the same results. Reproduced refers to the fact that perhaps if the experiment is repeated by somebody else, O in somebody, O in reproduced, they will get the same answer as I did. This, of course, will help to make the experiment more trustworthy. Double-blind testing also means that experiments will be fair. Double-blind testing is where neither the tester or the patient getting a trial drug perhaps knows what treatment is being given. Double-blind testing is used to eliminate any conscious or unconscious bias. So, for example, if the experimenter or doctor is giving a trial drug to patients, and he or she knows who is getting the active drug and who is getting the placebo, then the doctor could unintentionally give clues to the patients. A placebo is a substance with no active medication, and it is used as a control in an experiment. This bias leads to unreliable results because the patients receiving the real medication could claim that they felt better, whereas in actual fact the medicine might not be having any effect at all. Sample size is another important consideration when designing a fair experiment. This refers to how many items or persons are included in the experiment. In general, the larger the sample size, the more accurate the experiment will be. If the sample size is too small, individual differences among the subjects being tested may give inaccurate results. So, for example, when I want to investigate the average height of a student in my school, taking only 10 students will not give as accurate a reading as taking 500 students. Safety is another important consideration when undertaking scientific experimental work. You are already familiar with the guidelines for lab safety in school, and the guidelines for lab safety should be followed in order to work in a safe and organised manner. You will have already met the idea of tying back long hair, wearing lab coats, wearing safety glasses, avoiding contact between electricity and water, being aware of the safety information when using chemicals, etc. Thirdly, experimentation involves careful planning and design. During an experiment, only a single factor or a single thing is allowed to change. All other variables should be kept constant. There are 3 types of variables involved in a scientific experiment. The first type is the independent variable. Now, the independent variable is the one that you change during the experiment. Think, I am in charge of it. I for independent, I for I. So let's take an example. Let's suppose I want to investigate if I change the temperature of tea, then more sugar will dissolve. In this case, the independent variable, the variable that I change, that I am in charge of, is the temperature of the tea. I'm going to vary the temperature of the hot water. The second type of variable in a science experiment is the dependent variable. Now, the dependent variable is the one that changes as a result of the change that you made. So the dependent variable here in this example will be the amount of sugar that dissolves, because that's going to change as a result of changing the temperature of the water. As a heads up, the dependent variable is the variable or the thing that you're going to measure. Second letter E in measure, second letter E in dependent. E at the end of measure and E at the end of dependent. If you have to draw a graph of results, you usually put what you measure, the dependent variable, up the side. U for up, U in measure, S and E for up the side, S and E inside. So you put the dependent variable up the y-axis if you're graphing, and the independent variable usually goes along the x-axis. So dependent up the side and independent along the bottom. The last type of variable involved in a scientific experiment are the controlled variables. The controlled variables can also be called constant variables or fixed variables, CON in constant and CON in controlled. These are the variables or the things that are kept the same during the experiment. In this example, it would be the same volume of tea each time, stir the mixture the same number of times or for the same length of time, use the same style mug each time. Don't get controlled variables confused with a control. An experimental control is the final concept that we're going to talk about. Now, an experimental control is set up for comparison purposes. It acts as a standard against which the results of the actual experiment can be compared. So in my example, where I was investigating the average height of a student in my school, then the standard against which I can compare my result for my school could be the national average height. That would act as a standard against which I could compare the height of the students in my school. There should only be one difference between the actual control and the experiment. I would advise that you would learn this idea of an experimental control very well, because we're going to meet it a number of times and they are quite fond of it in exams. There you have it. Now, the principles of study are to practice in a jotter. Now that we have reached the end of our lesson, have we achieved our objective? Can you understand and describe the principles of experimentation?